first speakers today will be Dr. Norma Labarte. Dr. Norma is a PhD. Her major is in veterinary medicine by Federal University of Fluminense. She's a professor at post-graduation program in bioethics, applied ethics, and collective health from FIOCRUZ. And she's also FIOCRUZ president's assistant. Thank you very much, Dr. Norma Labarte, for accepting our invitation. And the second speaker is Dr. Jorge Guerrero, a PhD, and his major is in veterinary medicine from Illinois University. He's also a professor at medical school at University of Pennsylvania, and he's a member of several societies: American College of Veterinary Microbiology, European College of Veterinary Parasitology, American Society for Heart, Filariasis, founding member of the Southern European Veterinary Director Conference. Thank you very much, Dr. Jorge Guerrero, for accepting our invitation. And I wish you a very nice presentation. Thank you.
Are we on?
Yes, you're on.
Okay, I can see both of you.
Right, we can see your slides.
Let's go back. Oh, okay, now I'm the first one. Okay, so Jorge and I are here today to talk to you about our great friend Dirofilaria. And we are very close friends because of that worm. So the worm is very important for me because it took me from Niterói to Rome. And it kind of developed my interest in research. And Jorge also introduced me to a lot of important people that are from the very important associations in the world. So that's when my great friendship with Jorge started. Okay, but today I'll talk to you a little bit about Dirofilaria, and he will talk about Wolbachia, which is a symbiotic association. And very poorly comparing, I kind of feel myself as Dirofilaria that depends on Wolbachia. So he's my Wolbachia. Okay, so thank you very much for the university for kindness of inviting us to talk to you today. I'll be talking a little bit and I'll try to leave more time over to Jorge. So as Patricia already told you, I was graduated from Federal University Fluminense, and then when I retired, I started working at FIOCRUZ, and nowadays I work more at Federal Rural do Rio de Janeiro than any place else. So changing institutions is kind of interesting because it kind of renews you. You kind of breathe new air. It's pretty much a good thing to do. Okay, to remind you all who Dirofilaria is this nematode where the females are pretty long. They may be as long as 30 centimeters and the males are much shorter. Females are much larger in length and diameter than males are. And they are, you know, around in Brazil for a very long time. Males and females mate. They produce their babies, which are the microfilaria, the first stage larvae. And first stage larvae is when the mosquito takes its blood meal, the microfilaria goes into the digestive tract of the mosquito. They molt, they change from first stage to third stage in the mosquito, and these are the infective larvae. So mosquitoes are very important. We must keep in mind that canines are the best hosts for diarrhea. Although cats, feline, can be hosts as well. They're not as adapted, but they do get heartworms. Please remember that in a dog, the worms can survive up to 7 years in a dog. In the cat, they will survive for perhaps 2 years. They Males and females produce many larvae, many microfilaria during their lives. In the dogs, when the host is a dog, we usually see many microfilaria that can survive for 2 years. 30% of the dogs won't have microfilaremia, but most dogs do have microfilaremia, but cats don't. So don't expect to be able to diagnose heartworm infection in a cat relying on microfilaria detection. Don't do that. You may find microfilaria in a cat, but that's not a good way to look for it. It's just accidental. A cat won't have microfilaria for a long time, but dogs will. That's why we find so many dogs harboring microfilaria. So what does Wolbachia has to do with Dirofilaria? They're symbiotic. Dirofilaria depends on Wolbachia. Wolbachia doesn't depend on heartworm. Wolbachia can be in many, many different organisms in nature, but Dirofilaria demands its right number of Wolbachia in their bodies to be healthy. A heartworm with no Wolbachia is a, let's say, sick worm. The worm doesn't feel well, its fitness is gone. To remind you about the life cycle, if we start with the microfilaria in the canine blood, it's the microfilaria, it's the L1, the first stage larvae. Mosquitoes will feed on that host. They will get their blood and the microfilaria as well. They go through the digestive tract. It will take like 10 to 15 days, and these L1s are going to change, molt into L1, L2, and then L3s. After that, when this is the mosquito's proboscis, and there is the infective larvae. We have one here and one right there. So here you can see how large they are. They're pretty large. If you imagine this is the proboscis of the mosquitoes, these larvae are large. Okay, when this mosquito goes and takes his next blood meal on the dog, these larvae kind of swim out of the mosquito mouthparts into the canine skin, and then they're going to molt from L3 to L4, and that's going to take a couple of days. This is when these larvae are more sensitive to the preventives. All these macrocyclic lactones we know, they act better right here when the larvae are in the subcutaneous. Then it takes a couple days for the larvae to kind of find its way, turn into the juvenile and find its way to the thorax. And then they get into the heart and lungs. And then approximately 180 days later, we have microfilaria in the circulation again. So that's pretty much the life cycle in a dog. In the mosquito, this is very interesting because we don't have very many studies looking into this. And I think that's pretty nice part of the life cycle to think about. Mosquitoes will get their blood meal with microfilaria. Microfilaria and blood are sucked up to the digestive tract of the mosquito, and they have to go all the way in to complete their cycle in the mosquito. But this is the first problem the larvae are going to have to deal with. This pump right here may have a couple of teeth that will disrupt some larvae, and it can even disrupt all the larvae this mosquito is feeding on. So this is the first problem for the larvae. Many species of mosquitoes will have teeth, others don't. So this is a problem for the heartworm larvae to face in the mosquito. And then they go all the way through the digestive tract, and they have to swim to the tubules, the Malpighian tubules of the mosquitoes. They have to go in there. And this is very important for the larvae. So these mosquitoes, if you think of Culex mosquitoes, they don't have these teeth in this pump. But, I mean, these Culex have the teeth, and Ocrerotatus and Aedes, they don't have teeth inside here. So these are eventually the best group of mosquitoes because they don't have teeth in here. So larvae are going to go through here without being disrupted. But then mosquitoes are going to get more larvae than the Culex mosquitoes. And just as a reminder, mosquitoes may die of heartworm as well as dogs do. So this is the first problem. Then mosquitoes larvae have to swim inside the Malpighian tubes, which are 5 different tubes. They're dead end tubes. They go nowhere. They're 5 tubes. And so let's pretend this is the tubule, and this kind of orange organism in here is the larvae. So the larvae goes in there, has to swim all the way in. Then they have to go inside the cell. They have to get in the cell. And this is one cell line, only one cell. So it goes in there, it stays there, it gets shorter, stays there for a couple of days, then it goes out to the lumen of the tubule again, and there it makes 2 modes. It modes from L1 to L2, from L2 to L3, and L3 is very active. They're nervous larvae. They get very active, they go crazy, and they disrupt. Usually they go this way and they disrupt the tubule right there at the end. And then they start swimming backwards, and get to the mouth, to the head of the mosquito and stay close to the mouth parts. They don't get into the saliva or anything. They stay in the mouth parts. And when this mosquito gets its next blood meal, larvae then infect a new host. Please remember that this is an active infection. These larvae do swim and move across the mouth parts and get their way through the host skin to the subcutaneous. It's active infection. It takes, in the mosquito, something like 10 to 15 days. It depends on the temperature, environmental temperature. The warmer the climate, the faster the cycle, which makes Rio de Janeiro a very good place for the development of a infective larvae. Please remember that cats can be infected as well. The cycle in the cat's a bit longer, and usually they don't have as many adults as the dogs do. But remember, only one adult Dirofilaria is enough to make a cat sick and even kill a cat. So cats are more resistant than dogs are, but if they get infected, their life is in danger. And we can get our share of heartworms as well. We'll have a nodule in our lungs, is a pulmonary nodule. Sometimes you undergo surgery, find a piece of adult worm there or juvenile worm, and sometimes you can see it in image as well. Usually it's only one nodule in a person. Okay, remember we have Dirofilaria, whose nickname is heartworm, but it's not a heartworm, it's a lungworm. Okay, we'll find them sometimes in the ventricle, left and the right ventricle, but they rather be in the pulmonary artery, much better than the heart. We usually in necropsy see things like this because the worms, when the dog dies, they swim backwards to the heart, but they don't like to be there. They like to be in the pulmonary arteries. This is a pulmonary arteries with 120 adult worms or so. They really, really enjoy being in the lungs. Canine disease depends upon worm burden. The higher the number of parasites in a dog, it tends to be worse. The disease tends to be worse. But remember, the same 10 worms in a small dog means much more than 10 worms in a large dog. So that we have to consider that. The duration of infection, a long time infection may be worse than a recent infection. Also, we have to consider the individual reaction. Inflammatory reactions are the key for the disease. So if you have a dog with an individual inflammatory reaction that's very active, you have a worse disease. And physical activity. A dog that stays in the lap all day or in the sofa couch all day, the disease tends to be milder than the hunting dog. Most of the trauma heartworms will elicit is mechanical. They don't get fixed anywhere. They have to swim during 7 years against this very strong blood flow right here. So the mechanical endothelial aggression that happens inside the vessels is key. It's the first thing that happens. It's a trigger. And then all the inflammation happens and all these are formed, and we have lots of inflammation inside the lumen of the vessels, and we have inflammation, villi formation, and then the disease starts. To help on this inflammation, to make it worse, comes Wolbachia. Wolbachia is always there. It makes the worms healthier, but it also has an inflammatory capacity that makes the disease worse. So when a heartworm in an adult worm dies, all the Wolbachia that comes free from that worm is a problem, because it will elicit an inflammatory reaction worse than it would be if the Wolbachia weren't there. So the pressure problem goes back from the artery, from the pulmonary artery to the ventricle, to the atrium, and to the lung vessels. So it goes backwards. Okay, clinical signs, increased lung sounds. That's for us vets to find. Cough, that's what the owners will probably ask you, that's, tell you that something is wrong with their dogs. This is what the owners usually identify as a sign of disease. Heart murmurs, that's for our vets to find, tricuspid and pulmonary valves. Exercise intolerance, most of the time owners think their dogs are getting older. So don't expect them to come in and tell you the dog is kind of lazier because they think it's older. Jugular pulse, sometimes the owner will notice it, but it's rare. But when you find it, it's pretty interesting to see. Ascites and syncope are two things I don't see anymore. I used to see many years ago. Now I kind of don't see it any longer. I think we are diagnosing it faster, earlier. Okay, take note that platelets aggregate. Heartworm patients, they do have nervous platelets. Sometimes people see dogs with thrombocytopenia and think of Ehrlichia, and it may be heartworm. It can very well be Dirofilaria. Notice this thrombus here. This is why this dog died. See? Because platelets are very nervous. And I kind of tend to think that's something to do with the aggression that these worms will elicit, and to Wolbachia as well. There's a lot of information. This is an area of science we can do a lot of different studies still. It's pretty new, and I think we have a lot to learn. Okay, we have lots of papers on that. Please remember that chest rays are very important to evaluate how bad the disease is. Remember that this is a lung disease. Everything starts with the pulmonary artery. So x-rays, chest x-rays are very important. please note that this lung is pretty bad. So remember, we call it heartworm, but the first thing we see is a bad lung situation, okay? Echocardiogram will, most of the time, when the number of parasites is considerable, we can see these parallel lines are white, bright white, and they should be no larger than 1.3 centimeters. That's the largest size of a heartworm. If you find something that 2 lines white, brilliant white, they're like 2 centimeters wide, it's probably something else, not heartworm, not Dirofilaria. So how do we treat this? We have to think we need to increase clinical conditions, that most of lesions in the lungs are probably scars. So we're not going to make it any better, but we can make it, not to get worse. Eliminate all the forms of the parasites and avoid complications. And that's pretty hard to do, because when you kill the worms, you have all the Wolbachia coming from the inside to the outside, and you have thrombi everywhere. So please take note that adulticidal treatment, although it can be done, it has to be very cautious. The internationally recommended treatment is your arsenical drug. We don't have that marketed in Brazil. It's not in market anymore. It has been, but not anymore. So we don't have that. There's a surgery that's possible to be done, although it demands special equipment and special forceps, and you have to be very well trained to do that. Remember, you're going through the jugular vein, which is rather easy to dissect the jugular vein, but you're going all the way inside to the heart, and perhaps if you have the right equipment, you can go all the way to the pulmonary arteries and grab the worms and bring them out. But don't think that's easy. If you look in internet, Dr. Google will show you a lot of videos on that, but really be cautious. This is a dog that was sent from the University Federal Rural of Rio de Janeiro to Animalia. The dog was very sick, was almost dead. Well, they took care of the dog and they did surgery, and they got, I can't remember the number of adult worms they took out of the heart. And this is 12 hours afterwards, after the surgery. Remember, the dog wouldn't stand, wouldn't eat. And then the dog, now it's more than a year since the surgery was done, and the dog is still alive and well. So it's possible, but please don't think it's simple. It should be done by someone who knows what they're doing, and especially what they're not supposed to do. There are many things you're not supposed to do. If you break one of these worms, you're probably going to kill a dog. And if you're too rude, you may disrupt the inner structures of the heart, and you kill the dog. So that's pretty dramatic. It's possible, but I wouldn't do it. Okay, adulticidal treatment. In Brazil, all we have is what we call alternative treatment. It's made with doxycycline and macrocyclic lactones. Doxycycline, because we know that when we diminish the number of Wolbachia organisms in a worm, this worm will be kind of sick. It won't be good, So this is why we use doxycycline, not because doxycycline kills a worm, disrupts microfilaria. That's not the idea. The idea is to reduce the number of Wolbachia organisms in the worms in order to make the worm less fit. So if I don't have Wolbachia, I have a Dirofilaria immitis, that fitness is gone. Then the macrocyclic lactone come along and disrupt these worms. So that's why we always use both things together. Don't think that if you use only doxycycline, you're going to do it. You won't do it. If you use only macrocyclic lactones, forget it. It won't work. You both drugs working together. Okay, and with that, I change gears to Dr. Jorge Guerrero so he can tell you about the wonders of Wolbachia. And if I can suggest to the students, this is a marvelous field for you to explore and work and have your thesis and dissertations and everything on Wolbachia, because Wolbachia, to my impression, is something that if I were 25 years old, I would, grab Wolbachia and study it hard, because I think it may tell us a lot of things about in the future. So Jorge, please take over from now.
Thank you very much, Norma. As always, it's a pleasure to be with you, and it's an honor to follow you. I need to get to be able to share my slides. Could you please? Okay, good afternoon, colleagues. It's a pleasure for me to be speaking to you. I would have preferred, obviously, to be in Seropedica and be able to share with you directly in person. But unfortunately, this year has been so crazy. And due to the pandemic, we are all trying to accommodate the best we can. I also would like to congratulate the Universidad Federal Rural de Rio de Janeiro for putting together this beautiful idea of a meeting dedicated to graduate students. I think it's a huge opportunity for a graduate student to have these presentations. Anyway, what is Wolbachia pipientis? It's usually referred to and known only by the generic name, Wolbachia. It's a bacteria from the Rickettsia family, a close relative of Ehrlichia, Anaplasma, and others. It is gram-negative, and it's transmitted by it's an intracytoplasmatic bacteria transmitted transovarially. No horizontal transmission. Now, this bacteria is widespread among arthropods especially, but a few species of worms also serve as hosts of Wolbachia. It was Hammerstein et al. in 2012 estimated that Wolbachia is present in over 40% of insect species. And it is recognized that Wolbachia manipulates the reproductive efficiency of the insects. And this is done by an effect which is basically epigenetic. Just to remind you, epigenetics is the study of how your behavior and environment can cause changes that affect the way your genes work. Now, Wolbachia induces effects in insects that include male killing and also feminization and parthogenesis. And most notably, what they do is produce cytoplasmic incompatibility. This allows Wolbachia to rapidly spread in insect populations because infected females produce viable progeny, regardless if they mate with Wolbachia infected or non-infected individuals. While when a non-infected female mates with an infected male, no progeny is produced. And as you can see, there is a modification in the reproductive potential of the individuals infected or not infected with Wolbachia. Now, as we recognize, then Wolbachia in insects inhibits or modifies reproduction. In this little chart, you can see in a graphic manner what this concept is all about. But this, again, in insects. These effects are seemingly due to, again, by epigenetic effects. So this is an endosymbiont. The reason they have also some positive effects on the insect. So the insect serves as a host. The Wolbachia facilitates certain aspects of the physiology of the insect. Now, when you see them by electron micrograph, as you can see in this picture in which Culex pipiens, mosquito eggs, you can see them as these dark points. Wolbachia pipientis encompass a group of alpha-2 intracellular proteobacteria, which we recognize they are rickettsias. Now, as I said, they are mainly present in arthropods, but a few species of nematodes can also serve as a host for Wolbachia. And among them, our friend Dirofilaria immitis is one of them. But Wolbachia has not been recognized readily. It's only in 1924 when Hertig and Wolbach first described and identified this rickettsia-like microorganism in Culex pipiens. Hertig formally described the species in 1936 as Wolbachia pipientis. But as you can see in this chart, the knowledge on Wolbachia has been modified since 1924. Initially, Bill Kosick, at that time at Tulane University, recognized this as what he called inner corpora, considering that it was a micro organ in the microfilaria. Later on, Japanese investigators found this an unusual appearance. Some people recognized them as mitochondria, and it was only in 1975 that they were recognized to be bacteria. And later on, it was not until the mid-90s when it was really described and recognized as a bacteria, Wolbachia being recognized as an endosymbiont of Dirofilaria immitis. This paper by Cironi et al. Describes the molecular evidence for close relative of the arthropod endosymbiont Wolbachia in filarial worms. So as you can see, the recognition of Wolbachia in nematodes was not as simple a line. It was recognized as different things in different moments of the evolution of our molecular sciences. Wolbachias are present and recognized in the hypodermis of nematodes, mainly. They are found especially on the lateral cords, accumulated and easily recognizable by different stains. You can see this, the large accumulation of Wolbachia on the lateral cords of these nematodes. They can also be found in the rachis. It can be found in immature stages, in precursor cells, and in the oocytes, which is a way for them to be transferred from one generation to another. They are present in more than 17 species of nematodes, among them Dirofilaria immitis, Dirofilaria repens, Onchocerca volvulus, Boquereria bancrofti, Brugia malai, and other species, and Manzanella. But what's interesting is, even though Wolbachia is an endosymbiont of arthropods, you do not find it in the mosquito vector. Another interesting point, which Dr. Labarte already called attention, is that these are susceptible to doxycycline. Now, you can find this transfer from one generation to another of filarias in vertical transmission. You can see here the presence of Wolbachia in the reproductive organs of Filaria. This parasite, or this endosymbiosis, has developed over millions of years. There has been a coevolution. Phylogenetic studies in Wolbachia have shown that this phylogeny corresponds to the phylogeny of the nematode host. In addition, Wolbachia obtained from different isolates of the same species show sequences of identical genes. And that gives us an idea how old this close relationship is. There is evidence from phylogenetic studies that suggests that Wolbachia-Filaria association is one obligatory and species specific. So this tells us that the relationship between Dirofilaria immitis and Wolbachia is very, very close. The transmission of Wolbachia in nematodes, as I indicated before, is vertical. The two partners co-evolve for a long time. Now, Dr. Labarte gave a very clear description of heartworm infection in dogs. The old paradigm of this disease was the recognition that this was a disease caused by the filaria and the relationship with the vertebrate host. It wasn't until 1995 when it was this concept starts evolving and recognizing that the paradigm of this disease is the relation between filaria, Wolbachia, and the vertebrate host. But what does Wolbachia do to the parasite, to Dirofilaria immitis? Wolbachia facilitates the reproduction of filaria. Its effect is on the oogenesis, embryogenesis, and production of microfilaria. There is experimental evidence that indicates a positive effect of the Wolbachia on the molt between the L2 and L3. Remember, this happens in the mosquito. So at that moment, there is a very important participation of Wolbachia facilitating this molt between the L2 and L3, which makes this larvae infective. There is also a favorable effect on the molt once in the vertebrate. Once the L3 has already been introduced into the dog or a cat, the molt between the L4 and L5, which is a very critical point in the development of the worm, is facilitated by Wolbachia. And the hypothesis is that Wolbachia, what it does, how it helps, is by interfering with the host immune response. This is a very critical time, as I indicated, in the life cycle of the parasite. So the effect may be due to the production of antioxidants or catalase by the bacterium, or by changes in the network of cytokines that promote the survival of parasites. Then Think about it. Wolbachia then, at this point, is critical for the development of the parasite. Wolbachia is released. It's released constantly. It doesn't remain only inside the worm. In fact, the infected host then come into contact with Wolbachia by different ways. Obviously, after the death of the parasite. Death happens by wear and tear. When the microfilaria degrade, they liberate Wolbachia. By pharmacological interventions, as Dr. Labarte mentioned, the use of an adulticide will destroy the parasite and there will be a huge release of Wolbachia. Through the uterus, constantly when the female is delivering microfilaria, there will be release of Wolbachia. And also through the normal elimination of secretions and excretions of the parasite. These ES antigens are released constantly, and with them, Wolbachia is also released. Now, Wolbachia then in the host will accumulate in different organs, and you can see them as these red points once these histological sections are stained. There are many stains that can be utilized to demonstrate the presence of Wolbachia. Now, Wolbachia is a foreign microorganism to the mammal, so it will be recognized as a foreign antigen. And the most recognizable antigen of Wolbachia is the so-called Wolbachia surface protein. It is recognized by sera from positive cats and cats experimentally infected with Dirofilaria immitis, also by sera of positive dogs. This protein was not recognized by the sera for negative cats or negative dogs before the experimental infection with Dirofilaria immitis. The reason I tell you this is because, as Norma indicated, in cats, it is very difficult to diagnose Dirofilaria immitis infections. And our colleagues in Italy at the University of Milan, Claudio Cenchi and his team, tried to utilize the Wolbachia surface protein to diagnose the infection of Dirofilaria immitis, especially in cats. In cats, we have a huge challenge to manage the infection because, as Norma indicated, just one single worm can produce the death of this cat, and it's very difficult to diagnose. So this is an area of clear necessity to continue active research. Wolbachia is expressly recognized then by the immune system of the filaria infected host. There are specific antibodies against Wolbachia surface proteins that have been shown in different other filarias. For instance, Brugia malai in monkeys, Buceraria bancrofti in humans, and Manzonella in others. So Wolbachia is recognized by an infected host. Now, what does it do? What does Wolbachia do from the pathological standpoint? There is a lot of data indicating that Wolbachia plays an important role in the pathogenesis of the disease. It is causing inflammation. Dr. Labarte indicated when you kill the adult worm, it breaks down and releases huge amounts of Wolbachia. That is what precipitates the severe inflammatory reaction that is a consequence of the adulticide treatment. Wolbachia also is recognized as desensitizing the innate immune response and modifying the cytokine pattern and polarization of the immune response. So it actively participates in the pathological effect of the parasite. Remember, there is a lot of bacteria, a lot of Wolbachia inside the parasite. So there is a release of abundant antigen at different moments in the life of the parasite. Wolbachias produce molecules that are involved in the pathogenesis of filaria, not only being inflammatory by themselves, they actively participate in this inflammatory reaction that involve neutrophils, macrophages that release tumor necrosis factor, interleukin-1, and nitric oxide. They also act on T lymphocytes, and the lipopolysaccharide derived from Wolbachias activate macrophages, and this release tumor necrosis factor depending on a toll-like receptor 4, as well as interleukin-5 and interleukin-6. So Wolbachias are heavily involved in the progress of the disease. There are molecules associated with Wolbachia that can act as molecular patterns associated with pathogens. These are called molecular pattern associated with pathogens, or MPAPs. In monocytes, they activate this toll-like receptor and facilitate the production of reactive oxygen intermediates, nitrous oxide, like production of pro-inflammatory cytokines, and an upregulation of the adaptive immunity co-stimulating molecules. Anti-inflammatory mediators were also detected in living and dead filaria. This seems to be contrary to what I was saying, but we are going to see that when the release of Wolbachia is in small quantities during the life of the parasite, these Wolbachia tend to reduce the inflammatory reaction. And in many ways, this may be part of the host-parasite equilibrium, in which Wolbachia is highly involved. While the parasite is alive, then, the slow release of small amounts of Wolbachia and its molecules, like Wolbachia surface protein, induce a control of inflammatory mediators. And this has been beautifully demonstrated by Ray Dillon in a study performed a few years ago, in which he, utilizing cats, demonstrated that the physical presence of living immature parasites suppress the activity of the so-called pulmonary intravascular macrophages, or PIMS. And this was demonstrated to be by the slow release of small amounts of Wolbachia or Wolbachia surface protein. Dead young adults, on the other hand, release Wolbachia in large quantities and initiate a major PIM-mediated inflammatory reaction, which may be the cause of the death of many of the hosts, or the feline hosts, during different moments of the infection. For instance, when the L4s arrive in the pulmonary circulation, many of these die, and they induce the heavy reaction that may kill the cat. And this is a syndrome which is associated to the presence of Dirofilaria immitis in the pulmonary organs. The death of adult parasites also activates the inflammatory activity of these PIMS. So you can see how very complicated this relationship between Wolbachia, Dirofilaria, and the host. But there is a beautiful equilibrium. When the parasite is alive, the small quantities of Wolbachia, reduce the inflammatory response. When the parasite dies, there is a release of huge amounts of Wolbachia, which induce a severe inflammatory reaction. Filaria modulates the immune system of the mammals, of the mammalian host, to promote their own survival and fecundity. Now, this may be also regulated by Wolbachia, the production of the immune system mediators that tend to mitigate the specific immune response to the parasite. That way, the Filaria parasite survives. So, there are mechanisms that facilitate the life of the parasite and mechanisms that reduce the immune response in the host while the parasite is alive. Now, as Norma indicated, fortunately, these Wolbachias can be affected by tetracyclines in general and doxycycline especially, and that can be utilized to manage the infection. In 1971, our good friend John McCall at the University of Georgia had a problem with his gerbils in the experimental colony. He found that they had diarrhea, and to treat this diarrhea of these animals, which were infected experimentally with different types of filaria, Litomosoides sigmodontis, Acanthocheilonema viti, and Brugia pahangi. So he tried to save this colony and utilize tetracycline. And this was the first time that anybody saw a negative effect of tetracycline on the nematodes. And it was really, he scratched his head for many years because nobody could understand at that time how a tetracycline could have a negative effect on a nematode. And it was not until the recognition of Wolbachia, the effect of tetracycline of Wolbachia, and the disruption on this equilibrium with the parasite, that it was really recognized. Now, you have many references, many papers that have been published on Wolbachia and its influence on the pathology and immunology of Dirofilaria immitis infections. I have them in my presentation, and I recommend that you read them, or at least scan them. It's interesting that, as Norma said, doxycycline can be used to remove Wolbachia. You can see here in the left, you can see a picture of a healthy worm. It is a transversal section. You can see on the right, a transversal section of a Dirofilaria immitis in a dog that has received a treatment with doxycycline to remove the Wolbachia. And as you see, the difference is huge. The architecture of these two worms is completely different. This is a sick worm. It's probably a moribund dog caused by the elimination of the Wolbachia in this nematode. This has been tested many times, especially, as Norma indicated, in cases where immiticide or melarsomin, the arsenical, is not available to be utilized by the veterinary practitioner. And in fact, the American Heartworm Society has a protocol of treatment specifically including doxycycline in those cases where melarsomin is not available. And what you can see when the treatment of doxycycline is done, you can see the negative effect of the doxycycline on the structure of the nematode. We have done studies at the University of Georgia, as well as in my previous University of Pennsylvania, in which we experimented with this combination of treatments utilizing melarsomin and doxycycline in parallel, and we demonstrated this positive effect and also what we call a synergistic effect between these two therapies, which you will see in different pictures in the papers that we publish in veterinary parasitology. I see that we are getting very close to the limit. I don't want to leave you without a few points. The intermittent treatment with long-term doxycycline plus weekly ivermectin, as I indicated, is highly positive. The lesions improve greatly after adulticide treatment, 8 months after the infection and initiation of the treatment with ivermectin, doxycycline, compared to melarsomin alone. And you can see that this effect is seen in different histological sections of these parasites. Again, you can see this in the papers that I make reference, and you will have time to review them. Now, I indicated there was a synergism between ivermectin and doxycycline. Seems to be, why is it better together? And in the study that we did with Dr. Stephens and John McCall in 1998, we saw that the treatment with ivermectin retains the ingestion or damages the intestinal epithelium of parasites. And that may be the reason why when you treat with ivermectin and doxycycline, you have a synergistic effect. Anyway, I would like to close just with a few remarks. The evolutionary studies confirm that the filaria have been housing Wolbachia for tens of millions of years. During this time, the bacteria have learned to provide necessary metabolites for the worm. In turn, the filaria have ensured the survival and transmission of the bacteria. There's a general consistency indicating that Wolbachia plays a central role in innate immunity and the pathogenesis of the disease caused by the filaria. Wolbachia manipulates the immune response of the infected host, therefore is capable of allowing long-term survival of filaria in immunocompetent individuals. These last 2 points open the question of the biological role of Wolbachia in filaria. And this is very important for you to keep in mind. Now, just before leaving you, I would like to leave you with some questions that you as graduate students may take for your potential research. Are the Wolbachia complementary, eliminated after 1 month of doxycycline, or is it further time needed? And so on. Here you have the questions. I hope you review them, think about it, and take them as your projects for your graduate studies. Thank you very much again. I would like to thank once more to the university for having invited me. Thank you. If you have questions, please don't hesitate. And it can be in any language you want.
Thank you very much, Professor. Yes, we have questions. In fact, we have a very interesting conversation in the chat. I'm going to give you a feedback. Professor Douglas McIntosh said, Wolbachia is cool, microbiology of parasites. And Professor Fabio Scott said that Professor Douglas loves Wolbachia. And he said, I prefer Rickettsia, but Wolbachia are extremely interesting. And so we had a few questions for you and for Dr. Norma Labarte. Professor Fabio Scott asked, is there any participation of Wolbachia in the phenomenon of Dirofilaria resistance to ivermectin?
Norma or I, either one. I would like to answer that. No, I don't think there is any relationship between the presence of Wolbachia on resistance. Now, I am of the side that there is skeptical about this resistance phenomenon. This is, in my view, a limited problem of certain regions of the United States where first there has been very bad management of the disease, and second, a huge concentration of mosquitoes infected that transmit Dirofilaria immitis. So, no, but the relationship with Wolbachia resistance, no, there's none.
Right.
Norma, have any opinion?
Well, I kind of disagree, but it's just a hypothesis. If Wolbachia is spread all over the subcutaneous of the worm, the tissue, and when you have less Wolbachia, the worms have trouble molting from L4 to L5 to the juvenile form. So sometimes I kind of think that perhaps Wolbachia could play an important role in the production of the new cuticulum of the worm. So instead of being the resistance, instead of being a problem in the worm DNA, perhaps the problem may be in the Wolbachia DNA. That's just something I keep thinking about.
Interesting for the graduate students to investigate.
Yes, absolutely. There's another question for Dr. Norma from Isabella Vilhena. Is there any perspective for new adulticidal treatment in Brazil?
Well, the person more closer that's closer to the industry than I am is Jorge, but no, I don't think so. Not in Brazil and not in the world. I mean, we have the arsenicals all over the world. We don't have it in Brazil, marketed in Brazil, because we had and we didn't use it. So if you don't use, the company just gives up trying to sell it. So we don't have that any longer. We used to, but we don't. And what we are doing is using moxidectin associated with doxycycline or ivermectin or. So a macrocyclic lactone plus doxycycline. But I must say, if I'm not mistaken, we have documented results with ivermectin and with moxidectin. I don't recall papers with selamectin or milbemycin, but their mechanism of action is pretty close.
If I can add to what Norma said, I would like to remind you that the last new molecule developed for as a parasiticide and by accident was ivermectin. I was involved with the development of ivermectin since 1984, and had the pleasure and an honor to work with Bill Campbell at Merck and Company in the United States. And this was a discovery, accidental discovery, in the search of an antibiotic. Ivermectin was not a parasiticide. It was in the antibiotic search. And it was a failure as an antibiotic. But fortunately, a very astute investigator, and actually Bill Campbell, saw that in the cages of these mice that were part of the screening for antibiotics, there were a lot of Cyphacia and other nematodes in the mix in the bedding. And then he thought, maybe this has an anthelmintic effect. Then he went to the anti-parasite screening, and that way it was found to be this magnificent molecule it is. Now, the others, macrocyclic lactones, are variations of the same theme. So nothing new since 1980. And the reason is very simple. To discover and develop a new pharmaceutical entity costs approximately $1 billion. And it takes approximately 15 years since the discovery to the moment the molecule goes into the market. For that reason, no pharmaceutical company in its right mind is working on looking for a better melarsomin.
Very, very interesting. We have some more comments. Professor Thais is saying, thanks, Dr. Norma and Dr. Guerrero. Excellent presentations. We actually have time for one more question. We have still 3 minutes. Professor Julio Fernandes asked, what are the opportunities for future research involving Dirofilaria and Wolbachia? Well, Dr. Jorge just said a bunch of remaining questions, but what do you think it is more important to seek now, to investigate, to research?
Norma?
Well, I would focus on Wolbachia.
It's quite interesting. That's a very interesting question because I was in searching the literature, I start seeing that there are some attempts to utilize Wolbachia as a biological control of certain insects. Now, is there any possibility to use that in nematodes? I don't know. Again, that's a question for the graduate students. But indeed, I agree with Norma. If anything, keep working on Wolbachia.
Very nice. That might be a key for the next Nobel Prize, right? Very much, Professor.
Well, I assume I'll be fertilizer by that time.
Right. And Dr. Norma Labarte, thank you very much. These were excellent presentations. And we will have a break right now and we will come back for 20. Okay. So see you guys in a couple of minutes.
Thank you. Bye-bye.
Thank you very much.